like Dr. Anshul already introduced that I am working as a research professor in the MSc National University in Chittagong. It is the south part of the South India. So during the presentation, if there are any doubts or questions, you can note down or you can ask in between, both is okay. So like we all know that mutations are always in progress for any evolution that happens in any organism. Like any dynamic change is the source of mutation that leads to the novel elements and new guidelines. And from the ages, we have used the selective breeding as a tool to make new plant varieties, and that has been the initial source of having the different plant varieties for the agriculture. So with the advancement of the science and particularly the discovery of the radiation and chemical intelligence in the 20th century, which led how to be useful to induce the random mutations in any organism into the plants. So in the 1920s and 30s, this radiation and chemical intelligence were used to make a mutant library of plants and to scale further for the useful traits. And later on, the many languages divided this all into four major groups. First, radiation and chemical intelligences, and before that we know that conventional plant breeding. And third part is like first part is the conventional plant breeding, second was the random intelligences using radiation and chemical intelligence, and the third part is the transgenesis, or we can say the transgenic breeding, in which we got to know or we develop techniques to introduce a foreign gene into the desired plant variety and make them useful for specific trait improvement. Particularly, we all know the beauty cotton, which is very commonly famous in India. And from the last 20 years, we have shown how tools that we can repurpose for the precise gene editing, particularly the three main tools that were earlier popular, that is Talent, Xellia, and now CRISPR. And in the meantime, there are some other community tools that have been used for the introduction of mutations that leads to the crop improvement. So when we compare the different aspects of these four approaches that have been used for the plant breeding and crop improvement, we know that why CRISPR is getting more boom, or genome editing tools are considered more advantageous compared to the other tools. Particularly, the time required and is quite less compared to the other approaches. Second aspect that we can say is we can precisely edit the desired genomic aspects that leads to the crop improvement. And third, in some way we can say, like in case of transgenesis, we have concerns for the transgenics, that is GMO, genetically modified organisms. But in case of the in case of the CRISPR-based tools, that aspect can be avoided by removing the CRISPR tool from our developed plant material. So when it comes to the molecular scissors, that is CRISPR-based tools, these technologies are collectively known as new plant breeding technologies. And when this CRISPR-Cas system or the other meganucleases, all these are based on the making double-stranded break in the targeted genomic region that leads to the error-prone DNA repair pathway, or if we are providing some template that can be inserted into the double-stranded break, and that is called HDR pathway. So these two major DNA repair pathways that are instrumental in getting the outcome of the desired genome-edited lines, particularly non-homologous end-joining pathway leads to the error-prone DNA repair pathway, and that leads to the gene knockout, which is the most common application of CRISPR-Cas technology. And this CRISPR-Cas tool was repurposed for the geoengineering by these two wonderful scientists, Emmanuel Charpentier and Jennifer Doudna. And they were the first to give the proof of concept in 2011, and later they were awarded Nobel Prize for this technology in 2020. So it contains mainly two components, Cas protein, and another is the stretch of RNA, which is called as single guide RNA. And when these two forms a complex, it makes a Cas9 RNA complex. And this protein RNA complex looks for the guide RNA binding region, complementary region in the targeted genomic DNA. And when it finds it, this enzyme opens this strand, and there are two domains present in this enzyme that makes the cleavage in the targeted DNA. And that ultimately leads to the error-prone DNA repair or other specific outcomes that we intended to do using other accessory proteins. So this is the basic that you might all aware of now, and it requires one specific recognition sequence which should be followed by this guide RNA binding region, which is a guide like a RNA guide, and which is called as a PAM, that is photospacer adjacent motif. And the most famous Cas protein is the SP-Cas9, and that SP-Cas9 has a PAM sequence NGG, which is the most commonly used Cas enzyme in the CRISPR-Cas technology. So there are many other CRISPR-Cas systems that have been later discovered in different bacteria that acts as a immune systems in those native bacteria, but that have been now repurposed for doing the genome engineering work, and that includes many Cas proteins, particularly this Cas9 and Cas12a, or also alternatively earlier known as CPF1. These two are the major Cas orthologs from the two different species, and each of these different Cas proteins have the different properties. They can recognize different on-site. There are some unique patterns of the cleavage that can be induced by these Cas proteins. So depending on our purpose, we can choose the particular Cas protein, but particularly still, we can say it's a lucky case. SP-Cas9 is still the most popular and has the robust activity and more applicable in many scenarios. And that's why using the SP-Cas9 protein for its two mutant forms, one is the Cas9 leakage and another is the dead Cas9. So in case of Cas9 leakage, either one of these domains, that is HLH or RUBC domain, are mutated so that it can make a single-stranded DNA break rather than double-stranded break. And another mutant is dead Cas9, in which both the domains were mutated. So by introducing the single amino acid change in those mutations in these two domains, we can get these two forms. And based on that Cas9, there are many other tools that have been developed. Particularly, we know the base editors, which can make very precise nucleobase change, that is A to G, C to T, and many other are possible. And using these Cas projects, there are additional tools, that is targeted random mutagenesis tools that we can use for the protein evolution. Epigenetic modifiers, we can change the epigenetic marks on the genomic packaging or genomic DNA package, and transcriptional regulators, where we can knock down or upregulate the particular targeted gene. And in case of prime editor, which is a comparatively newer tool, but it has a huge potential that we can precisely change short stretches of the nucleobases at a desired target region. So when we see the tools, like what unique advantage these CRISPR-Cas tools provide to the genome biologists or molecular biologists, like earlier we can introduce some foreign genetic DNA to just have overexpression studies. Now with this CRISPR toolbox, we can edit all layers of the central dogma. That is, we know that DNA is packaged, and this packaged DNA contains many epigenetic marks, and later it it need to be transferred into RNA, and that leads to the protein. So we can edit these all layers using this CRISPR-based tools. So this is also one of the main advantage of having this CRISPR toolbox. So let us come to the base editors, which I'm going to talk in the upcoming slides. That base editors are based on the Cas9 linkage or Cas9 del Cas9 form. It consists of additional fusion of deaminase enzyme, which particularly deaminates specific single or multiple base nucleobases in the targeted DNA region, and that can lead to the C to T or A to G. These two are the most common or famous base editing enzymes or tools, that is the CB and AB. And recently there are additional tools, like when we consider 4 nucleobases ATGC, and we see their conversion from one to another. There are 2 possible conversions that we can do, and using these different base editors, we can now do virtually all 12 base changes. So A to G, C to T, or vice versa, and in a similar way, other remaining 8 different base conversions can be possible. And we all know this single base change can lead to the codon change, or multiple base changes can definitely lead to the codon change and ultimately change the amino acid, and that leads to the polymorphism, or we can change the single or multiple amino acids that are giving some particular trait, or that are helping the plant to do something that is not possible with the wild type DNA sequence. So these base editors are not just like earlier in previous slide I showed, A to G or C to T. Currently, we can also do the glycosylase base editor, that is G to C is possible. And in case of dual base editor, we can fuse the adenine and cytosine deaminase into to the Cas9 at one end or two ends. And that can lead to the simultaneous conversion of A to G and C to T. So we can have a more diversification of targeted genetic markers that can give us a more advantage, or that can produce a mutant library, and we can screen for the beneficial traits. Recently, in case of the bacteria, or even in mammalian cells, this one more additional tool is developed using the fusion of polymerase, particularly T7 polymerase, and cytosine deaminase. So this tool has limitations, particularly it requires the T7 RNA polymerase binding recognition motif, that is the promoter, and that we have to install first in a targeted cell or in targeted plant. That may be not currently easy, but it gives the advantage that this polymerase can drag this deaminases, and by moving for the making the RNA, it changes multiple nucleotides and can use, and also leads to the mutagenic library or mutant library development. So these different base editor based tools have been already applied or finding its applications in bacteria, yeast, plant, mammal, and there are many more coming up. So this is just a summary that I would like to show, that it has found many applications and are being optimized for different organisms. When we say example here, this RPOB that we will discuss later also, this is the RNA polymerase beta subunit, and mutations in this gene leads to the antibiotic resistance. Similarly, these other genes that have been studied in different organisms leads to the specific trait, and that we are studying using the mutant library development by applying this CRISPR-based base editing tools. You can look for these examples later on that I have given the citation, or you can simply note down these genes and you can find its applications and original papers. So when we see, in the nature, there is no natural enzyme that can make A to G, that is adenine to guanine. So the development group has developed a tool using the RNA adenosine deaminase, that is TAT enzyme. They have evolved it in the laboratory that can work on the DNA. And using this TAT, now there are many new big products that have been developed by further making many mutations into this TAT protein that can now give us many different editing outcomes, like single TAT with many mutations can lead to the it can give us the C to T also, or there are some C to G also, or A to C. So this single base change can be achieved using the TADE mutants, and this library is growing more and more. So we hope we will have more TADE mutants that can give us all 12 kind of base substitutions, and that will be a huge advantage to make a precision editing as well. So let us come to some experimental aspects, like why I chose to develop a platform that was that in bacteria before using any tool in the plants. So when in 5 years back, we also tried to adapt the CRISPR tool, particularly base editing tool for the plants, but it had very low efficiency. So it did not show any editing, and it is totally different than doing the base editing in single cell culture and doing in the whole plant system. So we were not sure why it was not working. So to troubleshoot that, we developed a platform named as EDCC, which is a in vitro or in vivo rapid investigation of CRISPR components in E. coli. And let us come to one by one into the detail. When we want to have a successful genome editing in a targeted plant species, we need to consider some basic aspects. Particularly, we need to know the target site and its properties, and what tool we need to use is dependent on the outcome that we intended to get, and single guide RNA features that are critical to the successful use of the CRISPR-Cas machinery for the genome engineering. And Cas features, particularly Cas9 enzyme features, what kind of farm site is available in the target region, we have to consider that by selecting the Cas protein as well. And expression machinery, which is not much focused aspect, but really critical for the successful use of these tools in plants. Because if our CRISPR components are not expressed at particular time point, we will not get the editing outcome that we are expecting in our experiments. And delivery method, that is also we need to consider. But there are many other aspects that we need to consider when we are using the CRISPR tool. And from among that, guide RNA features and expression machinery is critical. Because if our guide RNA is not functional, nothing will work. So, and also when we see some more tools coming up in mammalian cell particularly, and those tools will be tried to optimize in other system organisms, they work very low efficiency, or simply further optimization is required due to the inherent properties that are distinct from the mammalian cell systems when we consider bacteria or plants. So if we have a platform where we can verify some of the critical features of the CRISPR tool, it can facilitate or we can support for application in a targeted gene for the genome editing. So when we consider plants, there are like RNPs, like in vitro assays we can do, protoplast culture or infiltration together. So like infiltration in plant species is not successful, and protoplast culture also needs thorough optimization and maintenance. It is also not successful in many crops. It is very effective in rice for some of the crops, but not It's like routine. It is not possible to use it routinely within labs. And in case of IRBs, it is negative, but it can verify very little aspects. It cannot show us how your CRISPR machinery is like if we are overexpressing it, we cannot verify that aspect in the IMP. So to considering all those aspects, we considered E. coli as a prime model system, and for that, in the earlier discussion, we know that now it requires main two components: Cas protein or Cas9 fused with other effector protein and guide RNA. So the expression of these two cassets is need to be functional, and that can be verified in bacteria. And similar parts or same parts we can use directly for the plant genetic transformation. So this concept that we named as ELCC, because we can verify the expression modules of the Cas9 or Cas protein and guide RNA in the bacteria itself. And to do that, when we try to test this system in bacteria, as we all know, the bacterial promoters and plant promoters are not same, and some of the plant promoters or most of the plant promoters doesn't work in bacteria. And bacterial systems are more prone to the cell damage and cell death because there is no well-developed non-homologous end-joining pathway, which is a very quick pathway, and it helps the cells to survive and repair the DNA damage very quickly. But in E. coli, that pathway is absent. So if we have a higher expression of CRISPR component, that cell doesn't grow. So the first challenge was to optimize the CRISPR component expression that does not kill the cell, and still we can have the rapid test of these components. So to test what are the different promoters that can really be functional in bacteria, we tested some plant promoters and some bacterial promoters. And among the different tested promoters, we found that the bacterial promoters are obviously functional in bacteria, but 35S promoter, which is one of the most commonly used actually it's a viral promoter, but that is used in overexpression of CRISPR components in plant. That is also functionally E. coli. And also when we say the properties of the most commonly used U6 promoter for the guide RNA expression, which has a highly conserved -10 and -35S components, which are critical for the expression of subsequent encoded part, and that also had and considering these highly conserved main promoter elements, we anticipated it should work in bacteria. Yeah. So when we test to test that, we use the Golden Gate cloning kit, to clone the different components in the expression cassette and further to transform to make final vectors for the plant genetic transformation. We have many kits available from the different sources. So we use the Golden Gate modular assembly, where we can use the BSA, BSMD1, and BPI1, these three restriction enzymes that cut the DNA outside their recognition sequence and doesn't leave any scar. So it can be scarless cloning. There is no leftover from the recognition sites of different restriction enzymes. And in the initial testing, we made a universal target acceptor module where we can clone any DNA that we want to check for the editing, followed by the superpole GFP, and that we can change every time we have a different target. And you can say this is the architecture of this plasmid vector that we were cloned for testing the PLCDA, which changes C to T in the targeted DNA region. And For the guide RNA expression, we checked both the promoters, bacterial promoter and this plant promoter. And we observed plant promoter is equally functional and active for the guide RNA expression. That is efficient safe to K conversion at different sites, and that can we can get the editing 100% editing in the targeted genomic loci. So when we consider the cytosine-based editor, which is PMCDA is one of the example, we fuse additional DNA repair component, that is a uracil DNA glycosylase inhibitor. My slides are not full screen now. Yeah, my screen is visible.
No.
Dr. Ashwin, can you help me?
You have to share it again. You have to share it.
Just we are doing Rahul. Just a minute.
Yeah.
Can you share it again? Your screen share.
Yeah.
Yeah.
Is it visible?
Yeah. Now it's visible.
Go to slide more. Yeah, perfect.
Yes. Okay. Yeah. Okay. Let's continue. So, using this combination of different parameters, we could establish like expression cassette and other factors that can give us idea if our expression cassette for the Cas9 or Cas9 fused different CRISPR tools are functional, and also our guide RNA is functional. So to extend that work, if it really can be used for other cytosine-based editing enzymes, we tested two additional CDE versions, that is EOCDA and ApoB3A. So these both also converts C to T, and like earlier Dr. Kutu from Qatar, he also explained that in case of base editor, there is an additional concept, that is an editable window. So when this deaminase enzyme is fused with Cas9, it can access the multiple substrates, but it cannot go beyond particular range, and that range is called as editable window or editable window length. And that is specific for each deaminase enzyme, and depending on our purpose, we can select a particular deaminase that is more suitable for our work. So these two additional CDE versions were also tested, and that also showed efficient base editing. Additionally, the ABA version, which was earlier shown to be very poorly active in bacteria, we used the most advanced mutant, that is AB8E, that showed us the efficient A to G conversion in targeted sites. And also one additional mutant that has been reported in only plants we tested in bacteria, that also shown the efficient editing for the A to G conversion. So, and also in case of the base editors, like guide RNA length, we know that in case of ST-Cas9 it is approximately 20 base pair. Of course, we can use 19 or 18 base pair also, and it's also equally functional. And we can extend its length up to 25, or we tested up to 30 base pair, because these deaminase enzymes are evolved to work only on the single-stranded DNA, so that it will not damage other part of the DNA. And that's why if we can open the longer stretch of DNA by providing longer guide RNAs, it can work in a longer editing window length, but it may show some shifting of the editable region. And when we tested this concept, we have found that longer single guide RNA length showed the shifting of the editable window from one side towards the opposite direction. So as you can see, in case of all these three deaminages, PMCDA, AB90 or EOCDA1, these all editing windows were shifted towards the opposite direction of palm recognition. So ultimately, we can change the editable window length depending on our purpose when we use different length of the guide IDs. Yeah. So when we want to check some functional guide IDs that may not work, so you can see the still we know not many things are unknown. Like we can say we know a few things, but there are still more things that we don't know. When it comes to the guide RNA functionality, particularly when we see this some guide RNA, it contains some inverted repeats that makes the holding of this guide RNA structure then inactive. That becomes inactive, so it cannot work. That cannot be functional in the in vivo conditions, or there may be additional features that we don't know, because when we test many guide RNAs, some of them show the functionality, some doesn't show any functionality. It means it doesn't work at all. So to check if we can verify these functional and non-functional guide RNAs in using this platform, particularly with base editors, we tested this 5 guide RNA, and as expected, inactive guide RNA shows very little activity even after using the high strength promoters, where we expect many copies of the CRISPR components are produced. But still, they did not show the editing up to a remarkable efficiency. And in case of the moderate strength promoter like 35S promoter, it did not show any editing. So this also possible, but there are limitations when we want to verify our guide RNA functionality using this platform. Particularly, all these CRISPR-Cas machinery-based tools depend on the DNA repair machinery. We can make a double-stranded break. We can introduce some base deaminations. We can make some epigenetic modifications. But ultimately, how the DNA repair machinery on that particular plant or other organism responds is the main is also one of the driving factor that gives us success or failure in genome editing. That we cannot verify in this system. And also in case of bacteria, it's a quite simplified genome. In case of the plants, from plant to plant, we know that there is a huge genomic complexity. Not just the packaging, we know there are polyploidy and many other factors that simply we cannot verify using this kind of platforms. Or even we have some platforms in plant system based on the protoplast culture or some transient infiltration assay that also has these limitations. If that species is not the native one that we are intended to make a genome editing, we can still cannot verify those aspects. My slides are not changing. Yeah, let us wait. Yeah. So coming to the next part, I will go through these slides more quickly. Now, this is the extension of what platform we have developed and optimization of additional tools. In between, I will introduce some of the concepts about the Cas variants that are also crucial. So we wanted to have a dual base editors where we can fuse the adenine and cytosine deaminase together into the single tool, and that can give us A to G and C to T. So that will allow us more diversification of the targeted genetic loci, and we can have a mutant library with different editing outcomes that we can use for the screening. So this platform was developed and using the ABCC, and after getting these different fusions, we found that this adenine cytosine base editor 2 version and 4 version were very effective. So just I will go through some of these data. If you have questions later on, please ask me and we can have further discussion about it. So in case of the SP-Cas9, we know it requires NGG pump, and we cannot find NGG at every loci that we want to read. So we need additional SP-Cas9 mutants, that is virus, or some Cas version that can give us the accessibility to different sites with different palm motifs. So in 2019, 1819, these two variants were published that were the mutants of the SP-Cas9, this SP-Cas9-ENG and SPRY. So the name itself denotes that it recognizes the NG and SPRY recognizes the R or Y. In today's this talk, I will just cover the NG, and this NG contained 8 mutations in the SP-Cas9 that they were screened for the recognition of palm site, which is just ENG instead of NGG. So this relaxed palm version we fused with dual acidity components, and when we tested this dual acidity NG based version, we have found that it also works effectively at different palm sites, NGG, NGA, NGC, and NGT in 4 different loci. But you can say that it also edited the guide RNA region, so that we can say as a self-targeting. And this is one of the examples when we are having a plasmid vector to test our system. This is the Cas expression or the tool expression, and this is the guide RNA expression. So we are fusing this guide RNA guide region with this scaffold, and that is also present in this region. So this due to the recognition of NG as a palm, this produced component also make the changes in this guide RNA region that we say as a self-targeting, and you can see this also edited in this guide RNA expression cassette. And this is also reported in plants. This was actually the first report where they shown that with SP-Cas9 NG, it also targets the TDNA region, where we are expressing our single guide RNA using the U6 or other promoters. It also makes the changes. So there is a secondary guide RNA, which is random, and it can be very, it will be not good to get the proper editing, and also it may reduce the efficiency and it may give further off targets. So to reduce that cell targeting, this it recognizes NGG, so this 4 U were changed to the C, and this binder region to this one, when it makes a scaffold, was also changed. Its complementary basis also changed, and this was named as a modified single guide RNA scaffold. This involves single guide RNA scaffold, and it really reduced the cell target. There was no editing in the, we can say, the tDNA region or the guide RNA expression cassette. So new Cas variants comes with some advantages. Particularly, we can have the targeting of the NG or different palm sites containing regions, but also comes with some other costs that we have to reconsider. So to test the applicability of these different base editing variants that were developed using the SP-Cas9 or SP-Cas9-NG, we targeted one gene, which is RPOB. It is the RNA polymerase beta subunit gene, and we know Mycobacterium tuberculosis. It is the homologous gene that is also present in Mycobacterium, and mutations in 3 different regions of that gene leads to the antibiotic resistance. Like earlier, the rifampicin was a very effective drug to treat the Mycobacterium, but due to the mutations in these 3 different regions in that gene leads to the antibiotic resistance. That's why rifampicin is no longer effective drug to treat this disease alone. Like that drug alone is not effective anymore. And this homologous gene in E. coli also leads to the similar phenomenon. That is the mutations in these 3 regions are reported to give the difference in resistance to equalize. So we tested our tool to evolve this gene by targeting different seven locations using this guidelines with different palm size. So I will just summarize this into the table. Here I am showing this one. Like earlier, all mutations were known to be the single amino acid mutations that leads to the antibiotic resistance in case of RPOB mutations. But due to the availability of this dual base-editing tool, we could map the mutants that contain multiple amino acid substitutions that also leads to the antibiotic resistance. Additionally, some mutations that were previously known also found in this screening. So, using the this kind of tools, we can predict and study what kind of mutants leads to the antibiotic resistance. So, coming to the additional Cas variants, on the basis of the SP-Cas9, there is an additional mutant group that is circularly permutated Cas9, which contains the shuffling of this CRISPR Cas9 protein into different parts and connected by linkers. So this libraries were generated for the SP-Cas9, and among that, these two variants of the SP-Cas9 named as CP1028 and CP1041 that were shown to have more robust genome editing. So, and also in case of the bacteria, in case of mammalian system, that were shown to have the broader editing window when they were fused with the base editors. So we tested that in case of using the three base editing enzymes in bacteria, and it showed the wider editing window with some of the tested sites and in case of AB and CB version. In both the cases, this circular permutant SP-Cas9 fusion led to the wider editing window length. And let us go to another Cas9 ortholog, SP-Cas9 ortholog, SA-Cas9, which recognizes linearly RRT form and which is smaller than SP-Cas9, which requires around 21 to 23 single guide RNA for the effective editing. And this SA-Cas9 one mutant is earlier reported to have the more relaxed form recognition. Instead of linearly RRT, it can recognize linear RRT. So this SA package Cas9, these three are actually the mutations that are found that were found to be giving this property, that is the flexible palm recognition. So we tested this SA-Cas9 for the base editing, and similar like circular CP-Cas9 variants, SP-Cas9 variants, SA-Cas9 leads to the more effective and wider editing window length for the base editing. I will not go into detail, but just this EOCDA1, which is a CD variant, that gave us editing window, which is quite remarkably broader. So this EOCDA SDK-based version can give us the mutation spectra around 34 base pair long. So we can evolve some genes in this with this length, and that can really give us excellent mutant material to screen further for some specific traits. Similar to the dual-base editor, we tested these different Cas variant-based BE tools for the antibiotic evolution with referencing resistance protein, that is RPOB. And we found additional mutations that were not found in our earlier screening with investigators or not very previously known. So this is not what we have discovered in our screening. So let us come to the plants now, as we have seen that most of the tools that earlier I had to optimize in bacteria because they were not equally or almost non-functional in plants when they came in the beginning. And after the evolution of those CRISPR components, there have been continuous improvement in the efficiency, and also that show more detectable editing efficiencies in plants. So, using this platform, currently we are developing some tools that are completely new, not reported in any system. And also now we moved our work to test this system in model plant system and also in in tomato. So I will not go through these things in detail, but as we know that single guide RNA itself is a critical determinant of editing outcomes. So initially I screened 7 guide RNAs. Which one is more functional for the simple CRISPR-Cas editing, that is intent frequency. And we found this guide RNA1 was very active, and that was used to test different bee cultures. And now we know that these tools are really effective in tobacco test. And further, we are testing these tools in tomato and Arabidopsis. So let us just, I will, before closing the presentation, this is one of the paper that we published for the tomato crop improvement, particularly for the viral registers, that is based on the payload gene mutation. And these were earlier known, that is the MLO gene, which is known to be present found in many crops, and that gives the powdery mildew fungal disease registers. So, yeah, from the initially tool optimization, new tool development, and its application, we work across the different aspects of CRISPR. Yeah, and we already know that there are now products coming into the market, and particularly the high oleic soybean oil. It's not actually a CRISPR product. This was developed using the TALEN technology. But yeah, this is one of the genome editing product that is already marketed in USA. And in Japan, this GABA content tomato, it's also already, they distributed seeds for free to check the efficiency, and now they have marketed this high GABA content tomato by screening the mutants for a particular gene. And there are different publications, research and review papers that you can find on the internet. You can communicate with me, I can send you for that, and you are most welcome to have any discussions. Before that, I would like to introduce and thank to the Professor J.N. Kim, who is a supervisor and lab head, and he, from last 3 years, he has established one CRISPR-based startup, Nullabio, and that company provides services to the scientists. Particularly, they provide CRISPR mutants that in Arabidopsis and tomato for targeted genes. And additionally, currently for the gene tagging also, we have, like, the company has started this work also. And I would like to thank all the well-wishers and mentors in my scientific journey, and I would like to open our session for the discussion. Any kind of questions and suggestions are most welcome. Thank you.
So thank you, Dr. Rahul. So you have elaborated the system, how we can validate the guide RNA using the bacterial system that you have developed in vivo rapid evaluation of CRISPR-Cas base editing system in E. coli, as well as you have elaborated various apart from SP-Cas9, some other Cas9, and how you have done the things from the E. coli and then validated in plants. So thank you for the detailed and elaborate lecture. So now from audience, if there is any question. So I will start, like you were telling about your in vivo rapid evaluation system. So, like I have not gone through your publication, like which vector, like in case of a plant we are using binary vector. So here, which is the background vector? Is it in PET expression vector, or what kind of a vector you are using for expressing in E. coli?
same TDNA vectors that we use for the plant genetic transformation.
Okay.
We use the same one. We can use the same for the validation in E. coli.
Okay, so same binary vector will function in E. coli.
Yeah.
Okay. Yeah. From audience, anything? So from online, one question is coming. I will just read. So while going for optimization of Cas9, so just a minute. So while going for optimization of Cas9, you mentioned about RNP. What is the delivery system you have used?
Actually, I have summarized that in table. I haven't used that one directly. But most people use RNP like simply they synthesize the RNA using the different kits, and they prepare the synthetic DNA components, that is target DNA, and they mix it and just simply they run the gene to see that one. I haven't used this in current work, but yeah, that way people use the RNP system. Or in case of the plant for the protoplast culture, yeah, they just hybridize and make these components, purified purified CRISPR-Cas protein, like Cas protein and RNAs, and they make a complex and they simply do the bombardment or some other method to deliver these RNP components into the protoplast system.
Okay, so Rahul, there are some more online question. How do CRISPR-Cas9, CRISPR-Cas12 and CRISPR-Cas13 differ?
CRISPR-Cas9 and CRISPR-Cas12.
Cas12 and Cas13. So can you elaborate on that slide? I think from audience someone is asking how these three differ. CRISPR-Cas9, CRISPR-Cas12 and CRISPR-Cas13. So
Yeah.
can you elaborate on that slide? Yeah.
So Cas13 I haven't drawn here, but Cas13 is RNA editing enzyme. It is not DNA editing. So here I have focused more on the DNA editing toolbox. But just for example, when we have a Cas12a, which is also known earlier popularly known as a CPF1, so it recognizes the 80 reached form, which is different than the SP-Cas9 or some other Cas orthologs, and it makes the staggered end cut. When it make a DNA break, it doesn't make on the same position on both the strands. It makes in a, just like EcoR1 make a staggered end cut, not the blunt end. So this Cas12 make staggered end cut. So it increases the probability of the intergeneration, but definitely it's less robust than the SP-Cas9. Yeah, this is the difference between these two. But when it comes to the Cas13, Cas13 is a RNA editing enzyme. It doesn't work on DNA, and that is mostly used for the diagnostic purposes, particularly to detect the pathogens or some contaminations in the different food samples or in the human system also. There are many kits, diagnostic kits that have been developed to make using the properties to use the properties of Cas13 protein.
Okay, so there is one more online question. Can CRISPR-Cas technology be used to enhance production of spirulina?
I can't be very specific about spirulina. I don't have enough background information about the species, but if there are some more, like if we know some components, its inhibition or the activation can lead to the production, definitely we can apply CRISPR-Cas tools for that purpose.
Okay, thank you. So many compliments are coming about very nice presentation. So from online participant, if there is any question, you can type in the chat box or you can unmute, you can ask. Okay, if not, we would like to thanks. There is one more question. Can CRISPR-Cas9 also be modified?
Yeah, it's already modified for many purposes. Like particularly SPCas9, we know that wild type SPCas9 is recognized as NGG palm, and it is already modified to recognize many other palm sites. That is one of the things that have been done using by making the modifications in the Cas9 protein, and similarly that the CRISPR compound engineering can be definitely useful to make CRISPR-Cas machinery with the new properties that with the new kind of genome editing we can achieve. Yeah, definitely it's already done and it's being done.
Okay. So if there is no more question, we would like to thanks Dr. Rahul for accepting our invitation and delivering online lecture from Korea. So thank you, Rahul. So whenever you are coming to India, please visit our center.
Yeah, sure. Thank you. Thank you very much for giving me this opportunity. It's a
Okay.
great honor.
And I would request if you can share your publication on that IRECCE which you have elaborated so that we can share that with participants so that they can have an idea how we can validate this guide RNA before we are going to plants. So that way we can share with all of the participants. So thank you,
Yeah,
Rahul.
sure. Yeah, thank
Thank
you.
you,
Thank
thank
you
you,
very much.
thank you. Okay.
Recording stopped.